All right. So welcome to the Collaborative Science for Estuaries webinar series. I'm Lynn Baccaro. I'm part of the team that runs the Science Collaborative, and I'm joined here with Megan Brass, my close colleague, and 3 members of one of our project teams that I'll be presenting. And we are really thrilled to have such a great turnout today. We had 239 people registered, and it looks like there's folks who are just joining here. But let's go ahead and jump in because these guys have got a full presentation for you planned for this hour. So just some quick background here on our program and then the reserve system that we work with really closely. So the National Estuarine Research Reserve System, which goes by the acronym NERRS, is a national network of unique research reserves that are shown here on this map. This is a NOAA program that works in collaboration with a local place-based partner, either a state agency, a university, or in some cases a nonprofit. And each reserve site includes programs focused on land stewardship, research, scientific monitoring, training programs, and education. And then the program that I work for is called the Science Collaborative, and we support science for estuarine and coastal decision makers. So we coordinate regular funding programs and opportunities for user-driven collaborative research, assessment, and transfer projects. And you'll be hearing about one of those collaborative research projects here today in a minute. So some quick housekeeping items for our webinar today. You all were muted when you joined the webinar today because we've got such a great turnout today. But you are welcome to use the Q&A feature to put questions for the presenters at any point during the webinar. You can even upvote questions that you like, and we'll cover as many as we can at the end of the webinar today. The chat is also open and it's visible to everybody. So you're welcome to add some comments there. If there's resources or things that you think other participants might be interested in, you can put them there. Or you can put technical questions that you want to address to us as the organizers. So we are going to jump right in then. I'm going to introduce our 3 presenters today who are all part of one project team that's been looking at oyster aquaculture and whether it can help restore coastal water quality. So Tonna-Marie is going to be kicking us off. She is the manager of the Waquoit Bay National Estuarine Research Reserve in Massachusetts, and she served as the collaborative lead for this project. Tonna-Marie has got 15 years of experience connecting science and management and engaging stakeholders in research and planning processes, which was a tremendous asset for this project. And then she'll be passing the baton to Dan Rogers. Dan is an assistant professor of chemistry at Stonehill College, and he served as the principal investigator for this project. And his research focuses on biogeochemical cycling in coastal and deep sea environments and the development of new tools to study biological activity, including some of the techniques that were used in this project for measuring nitrogen gas fluxes. And you'll also be hearing from Paraskevi, who also goes by Vivian, and she is a research associate at Woods Hole Oceanographic Institution. Vivian specializes in marine microbiology, gene expression, and biogeochemistry in ocean water and marine sediments. And you'll hear more about her contribution to this project in a few minutes. So those are the 3 folks we'll be hearing from. We are really thrilled to have all of you here today. I'm going to pass it to Tonna-Marie to kick us off and tell us a little bit about how this project got started.
All right, good afternoon, everyone, and thank you so much, Lynn, for that introduction. My colleagues and I are really pleased that you've decided to spend an hour with us this afternoon when we know so many things are going on and everybody's very busy. So thank you for your interest. I'm going to tee it up this afternoon by just giving a very, very brief overview of the motivation for this study, why we did the work and the Cape Cod context, which is where we did the study, why the study was relevant in this place. I'm also just going to give a nod to the collaborative research approach that we use and why we went that route, as well as the team that we assembled, which we're hearing from 3 of us today, but not the entire team. The target audience that we had in mind when we decided to do this work, because we always had our eye on the ball in terms of who could use the science in the real world in terms of making management decisions. And so I'll give you a flavor of who those people are. And then at the end, Dan and Vivian will talk about the science results, as well as give you a sense of the deliberations that we've had about the potential management applications of the work. And then we want to leave you with some ideas of project resources that we've developed to help you learn and dig into further about this topic. So to start and set the stage, I'd like you to just consider this. Imagine your coastal community and what you think of when you're there. Perhaps you think of a beach environment, someplace you love to visit with family and friends. Perhaps it's the coastal setting, just the scenery. Someplace like on Cape Cod, lighthouses are very special places. Or perhaps it's just taking a stroll down Main Street and sort of visiting small mom and pop stores and just being in that place and soaking that all in. There is something that ties all of these enjoyable sort of experiences and thoughts together. And it is the idea and the reality and the need to have a healthy coastal environment. But what we're faced with today is that human activities has really impacted our coast. I think that everybody on this line would probably agree with that and know that right off the bat. But to tee up our study, one of the things that it's important to understand is that there's a lot of intense population pressure on the coast. In fact, 40% of the population lives less than 100 kilometers from the coast. And this intense population pressure has led to human activities that have altered the coastal ecosystem, sometimes in very negative ways. And we're seeing impacts such as overfishing of many species or loss of habitat can be two of the impacts that you think of right off the bat. But there is one other threat that is also in focus and was what we attempted to work on in this project. And it's the threat of nitrogen pollution. And nitrogen pollution has reared its ugly head, unfortunately, in many of our coastal communities on Cape Cod where we did this study. And communities have been grappling with this issue for a long time. They're still grappling with it today. And on Cape Cod, we have most of our homes and businesses are on septic systems. Not everywhere is sewered. And these septic systems do not do the best job of removing all the nitrogen that they possibly could. And so when the nitrogen enters the coastal environment, it fuels a cascade of negative effects. Some of these include decrease in available oxygen, decreased biodiversity. All these things are connected. And you can have detrimental impact on the economy. And for a place like Cape Cod, where it's a very tourism-centric economy, you can imagine that what's happening on the environment and what's happening in the economy are very interconnected. And so it's within this context that I would like you to just step in with me into where our project began. And the story is a Town of Falmouth, a local community, represents one of the 15 communities on Cape Cod that are dealing with this issue of nitrogen pollution. And the struggle is to find solutions that will be effective but will also be something that communities can afford. And so we're looking for cost-effective solutions that we know can address the problem. And so communities are under pressure to both find solutions that will work, find solutions that they can afford, and find solutions that will be effective in the long term. And so our study, many communities began to look at the issue of, can we use some non-traditional methods to address the problem? And by non-traditional, I mean not something as soaring, which is sort of a tried and true technology, but has a lot of cost involved and has issues related to water and land availability that will also have to be solved. We'll look at some other methods that communities could use. And one of them became a very popular, you could almost say a hot topic, and it's the topic of oyster aquaculture, with the understanding that we know that oysters can great filter feeders and can address this issue. But the question was, how effective could this particular approach be in the context of communities developing comprehensive water quality management plans and looking at targeted solutions that could work and that regulators could be pleased with? And so it's here that we began to address how can oyster aquaculture impact the nitrogen cycle. And today, what you're going to hear from Dan and Vivian, they're going to step through some of the key questions that were raised and around which we framed our study. But to help tee up the science, I'm going to ask Megan to help me bring up two polls. The first question, we just wanted to get a sense and engage you today in sort of helping to see how many people have an understanding of what oysters do in the environment. And so the first question I'd like to pose is, how do oysters help to promote the cycling of excess nutrients in coastal water bodies? The four answer choices are there. I'd encourage you to respond quickly so that we can get to the other poll and see what your results are. So how do oysters help to promote the cycling of excess nutrients in coastal water bodies? I'll give it a few seconds before we pull up the results. The four choices are, do they absorb nutrients from the water like a sponge? Do they eat microscopic plants that use the nutrients in the water? Or do they oysters absorb nutrients directly from the air, or all of the above? Megan, could you go ahead and just share the results you have so far, please? Okay, and so we have just about 50% of our audience that thinks it's the second choice, that they eat the microscopic plants, but we also have a healthy group that thinks all of the above, or that it is that they absorb nutrients from the water like a sponge. Now I'm going to leave you hanging with the answers, because when I ask my colleague Dan to pick up, he will answer that question and flow right into the presentation. So hold that thought in your mind. Megan, if you could close that poll and bring up the second one, please. All right. So this question gets right into the meat of what happens in the sediment as it relates to nitrogen cycling. So the question I pose is, do the microorganisms that remove nitrogen from the sediments require oxygen? And this is going to be a very important consideration for the rest of the talk. Please go ahead and enter your responses. We'll just give it another second so we can get as many responses as possible. Logged in. Megan, if you could go ahead and just share the results that we have. Aha, I see that just about 50% of our audience thinks that it's complicated. You know, one of those like, could be either way. Or we have a tie for yes and no. And again, I'm going to leave you hanging there because Dan is going to answer that question as we go through. So I'd like you to hold those two thoughts in mind as Dan picks up the presentation. First is what the oysters do in terms of how are they actually function in the environment, and then what happens in the sediment. And the last thought is, we're going to be talking about a lot of sediment chemistry today. Hang on, it's really important. And it's because of one of those questions that we really had to power through to get the answers to give science that could be applicable in a real world management context. So Dan, I'm going to invite you to pick it up here and take us through the nitrogen cycle and the rest of the story with Vivian.
Okay, thank you. So to address the poll questions first, do oysters take up nutrients directly from the water column? No, they take it up from their food source, just like we take up nutrients from our food source. Although sometimes it feels like we gain mass just by walking around, our BMI only goes up with what we eat. Oysters will eat the phytoplankton, or the microscopic plants that bloom because the nutrients are in the water column. Now, when they absorb these or ingest these phytoplankton, they can process some of them and absorb them into their own biomass, the same way you absorb the biomass from your foodstuffs. But some of them they don't want to eat. What oysters do that are kind of fun and interesting is the pieces that they don't want to eat, they package together in a nice tight little ball called a pseudofeces, and they eject that out of them. And so it's kind of like the picky kid segregating all the peas on his plate, forming them into a nice tight pea ball, and then throwing it back in your face. It's an efficient way to transform that inorganic matter from his plate to your face, or in the oyster's case, from the oyster gill to the sediments lying below. They sink them much faster as this dense particle than they would just dying phytoplankton. For the second part of the poll question, is oxygen required to remove nitrogen? You're right, it's complicated. The nitrogen cycle is a very complicated cycle with lots of intermediates, and we need both oxic and anoxic, or oxygen-containing and oxygen-lacking environments to process nitrogen. And that's where these sediments come into play. So we're going to take a quick tour through the nitrogen cycle, and there we go. And we're going to walk through what happens as we mobilize nitrogen from our organic matter and get it all the way back to dinitrogen, or N2, which makes up 80% of the breath you just took. So we're going to start at organic matter. And so hopefully you can see my laser circling this ammonium molecule right here. When we first burn our organic matter, we're going to produce ammonium. This is what's going to come out in your septic tank if you live in a place like Cape Cod, and it's the first product of that mineralization. If there's oxygen present, there are bacteria that will very quickly oxidize this ammonium all the way down to what we call nitrate down here at the bottom, only if oxygen is present. In a place like a septic tank or the grass on your front lawn, there is oxygen present, so this process happens relatively quickly. What's interesting about nitrate is that it's very mobile in the environment. If there's oxygen present, there's not a whole lot that can happen with the nitrate. There are a few species like plants that can take it up and put it back into biomass, but most animals can't do anything with nitrate. We can't do anything with nitrate. And so it moves quickly through the environment. In a place like Cape Cod, it's going to enter the groundwater system and it's going to move through the groundwater system until it comes out onto the coastal waters. Once it hits the coastal waters, the phytoplankton, the microscopic plants, are sitting there waiting to take it up. They're often limited by the amount of nitrogen that is available to them, just like your lawn is limited by the amount of nutrient available to it. And so that can stimulate this growth. Well, that's if it's in an oxic environment. If we can get this nitrogen into an anaerobic environment, there are other processes that take over. The first process we want to talk about is something called denitrification. And again, this is all happens in the absence of oxygen. In the absence of oxygen, there are lots of bacteria that can use nitrate the same way we use oxygen to burn their organic matter. And to do this, they're going to step nitrogen through a bunch of intermediates, nitrate, nitrous oxide, and ultimately to nitrogen gas or dinitrogen gas. And this gas is what we want to produce. This gas will evade from the ecosystem back into the atmosphere where it's virtually inert and not biologically reactive. That's where most of our nitrogen is going to be stored. There's another process that complements this called anammox. Again, it's an anaerobic or oxygen sensitive process, and it combines nitrite, one of the intermediates of this denitrification pathway, and that original ammonium that we get from remineralization of organic matter together to produce dinitrogen, again, resulting in the removal of this nitrogen from the ecosystem. The third process we're going to talk about today is something called dissimilatory nitrate reduction to ammonium, or DNRA, as it's easier to say. This is contrary to the first two processes. DNRA is an anaerobic process, but it results in the movement of the nitrogen in nitrate back to ammonium instead of out to dinitrogen. And this movement back to ammonium is important because it can lead to the storage of the nitrogen in the environment. All three of these systems taken together, these pathways taken together, there we go, form a fairly complex system of processes that can affect where nitrogen is in the environment. What controls these processes in part is what the other chemistry in the environment is like. Is there oxygen available? How much sulfite is available? How much organic matter is available? And they're all interlinked. Now, as a chemist, we can sit here and we can measure the amount of dinitrogen produced, and we're going to show you that in just a second. But with my biologist colleagues, we can also look at the different pathways a little bit differently. Each of these arrows represents a step in the nitrogen cycle, and we have marker genes for each of these steps. And so we can look for these markers of that process in the environment. And Vivian's going to tell us about that in a little bit. So our study was set up like this. We wanted to do a study that was meaningful to the growers, the managers, and all the different end users in our community. We didn't want to run the study as we, the scientists, might want to run a study. It wasn't going to be pristine. And so we partnered with the Town of Falmouth, and we went and asked one of the major fishing suppliers, what are the three most common types of oyster growth systems that are used in our area? And we had these three systems: a floating bag system, a midwater system called an oyster growth system, and a system of cages that sat in the bottom, and hence bottom cages. What these really do are change where the oysters are grown in relation to the bottom. We stocked all of these at the same density of oysters, and we asked the Town of Falmouth to manage these as they would any other aquaculture system. So it's managed under real practices. We put all these systems in the same, or as close as we could get to the same hydrographic setting at the head of Wuquoit Bay. Right here, you have all 3 systems plus our bare sediment control site. And what we're really going to do is we're going to look at the differences between our aquaculture systems and our control site to look to see how we're changing the system with this practice. So the big questions we're trying to answer is, does the aquaculture activity change the nitrogen release from the sediments? Do the microbial communities in the sediments change as a result of these aquaculture activities? Do their activities change as a result of their aquaculture activities? And is the amount of nitrogen released from the sediment enough to be included in management planning, water quality planning? So this is how we do it. We're going to tell you a story in 3 parts. I'm going to tell you about the chemistry and the nitrogen release from the sediments. Vivian's going to come and talk about the microbiology in the sediments, and then we're going to sum it all up in our lessons learned to take home. So that's where we're headed. How we did this is we set these systems up in that in the head of Wacoit Bay for 2 summers, removing them in the winter and setting them back up in the following spring. And we would go out to these sites every 2 weeks and sample. We would take cores in the central part of each of the setups, so we avoid edge effects, and we process those cores right on the beach. So we're getting sort of as close to real-time samples as we can. We monitored carbon data over the course of the growing season, and what we can see is that these bottom lines right here are from our control site in both years. We saw that the floating bag and the oyster grow systems, the mid-water system and the floating system, did put more carbon into the sediments than the control did. But really this bottom cage system put a lot more carbon into the sediments than the control sediments experienced. And so we're looking at something like a 14% increase in total carbon in the sediments. So we're seeing an efficient transport of organic matter to the sediments as a function of the oyster activities. We also did flux measurements of nitrogen across the sediment water interface by using core samples and measuring in real time oxygen release, oxygen flux across the sediments, and then a initial and final nitrogen measurement to calculate a nitrogen flux. And what we can see is that there is certainly a seasonal dependence on nitrogen flux as we go later into the summer, especially into August and September. We see this dramatic increase in nitrogen flux out of the sediments. But what we can also see is that our core treatments showed a significantly greater amount of nitrogen release than our control sediments did here in the blue. And that is consistent across both years. And if we start to play some games with numbers, we can sort of categorize our control sediments as inhabiting this sort of red zone in the last 2 months of the growing season. And we can calculate how much nitrogen was removed from the control sediments. And we get about 1/3 of a kilogram of nitrogen between August and September for that control plot. We can do the same sort of back of the envelope calculation for our aquaculture sites. And we can see we can get up to a kilogram, about 3 times more nitrogen out from each of these small farms. Now these farms are small, they're only about 250 square meters. These are not quite as big as a commercial farm might be, but that's a significant increase in nitrogen production over the course of the growing season. And so with that, I'm going to hand it to Dr. Vivian Mara to tell you about some of the biological markers of what is actually happening. What's the mechanism driving these observations?
So hello also from me. Thank you, Dan. So the question is, as Dan said in the beginning, all these processes of removing nitrogen from an environment, from the sediment, is happening via microbiological processes that they transform nitrate or ammonia into nitrogen gas. So it is very important also to have in mind that these processes are processes that they are happening naturally in the environment. So our basic question was to see which processes that Dan earlier described from the nitrogen cycle are affected in the presence of oysters, and towards which way, positively, negatively, and to have an idea of how this cycling occurs how this nitrogen cycle occurs with the presence of the oysters. So in order to be able to answer this question, the first thing we had to do is, first of all, do some DNA forensics, meaning that we had to be able to identify underneath every different oyster treatment that we used for this experiment, we had to find out which bacteria are present inside the sediment. This is the first thing that this was the first thing that we had to identify. So basically, what we did is we took samples from the sediment and we analyzed the DNA that it was present inside the sediment underneath the oyster cultures. And from there, we were able to find out the DNA fingerprint of every different bacterial group that it was present. The next question that we had to answer, of course, was, okay, now that we know which bacteria are present, which bacterial communities are present under the different oyster treatments, the question is what they are doing. So in order to be able to answer this question, we had to address, first of all, what it is called the whole metabolic pool, meaning all the possible metabolic processes that they are happening in the sediment in the presence of oysters, of the different oyster treatments that we applied. Of course, as we said in the beginning, our question was very specific. We didn't want to, at this point at least, at that point at least, we didn't want to evaluate the whole metabolic processes that the microbial communities were doing in the sediments, but rather to be able to address specific question. What about nitrogen? What's happening with regards to nitrogen? So for this reason, we had to isolate from this, let's say, big pool of information regarding metabolism. We had to isolate only those genes that they were related with nitrogen, and they would be able to explain to us more specifically which processes are present and under which different treatment. So, as we can see here, these are like here we have two plots that corresponds to 2018 and 2019 respectively. And here on the left side, you see actually all the different oyster treatments that we had. And at the same time, as we can see here, is the different genes, marker genes that we used in order to be able to quantify how denitrification changes underneath the different oyster treatments. So with the bubbles that you see here, everywhere that you can see a bubble, that means that the expression of this gene was higher when compared to the control. Where you see blank spaces like here or here, that means that the expression of these specific denitrification genes compared to the control was negligible. As we can see in some cases, the actual denitrification enhanced a lot, especially after the end of July. And this happened in both sequential years. And happened also not only in one gene, as we are seeing here, but also in the rest of genes, but of course not in the extent that this specific group had. So if you remember from the previous slide that Dan actually showed you, this was the first hint that the nitrogen production that Dan was able to measure that was released from the sediments underneath the different oyster treatments was somehow showing the same pattern with the genes that they were responsible for the release of this nitrogen in the atmosphere. The next thing that we had actually to answer, as you remember, is that Dan mentioned about other processes that they can also help nitrogen release from the sediments to the atmosphere due to the presence of oysters. So in this case, we targeted another gene of interest that it's a marker gene of Anamox. So as we can see here with blue, again, we are presenting data for 2018 and 2019. So as we can see here with the blue color is again the control. As you see, the Anamox as a process occurs during both sampling years. But what it is interesting is that the bottom cages in specific, which is actually the oyster treatment that has the closest proximity to the sediment, is the one that, as we can see, increased the expression of the marker gene that has to do with Anamox. That means that the bottom cages had higher release of nitrogen compared to the other treatments, like the oyster grove, the floating bag. So, however, please do remember that every different process in the nitrogen cycle that we are going to discuss here does not contribute in the same percentage in nitrogen release. The primary source for nitrogen release in the environment is denitrification, and then to a lesser extent, like a 10 or 15%, it's Anamox. Also, Dan will talk to you later about this very beautiful drop that we saw in both years when it comes to the oyster growth, and he will actually explain to you very beautifully what means presence of oxygen in a total anoxic situation. And the last thing that we had to discuss, of course, if we wanted to be fair and not be biased from our findings, was okay. We found we have studied two pathways that they are dealing with nitrogen release. However, in the beginning of the talk, Dan mentioned that nitrogen cycling also involves a step, when the conditions are appropriate, toward retention of nitrogen in the ecosystem. And that was something called DNRA. So if we had to be fair about this project, we also had to make sure and see what percentage of nitrogen retention we are having in the ecosystem because of the presence of the oyster. So just to explain also something here, as I said, all these processes that they happen are proportional. And in order for us to make, to have a solid conclusion, we had to budget all these fluxes and everything that we were calculating in order to have a fair and square outcome when it comes to nitrogen release. So as we can see here, both in 2019 and 2019, DNRA as a process, meaning nitrogen retention in the ecosystem, is a physical process that was happening in the ecosystem. So that's why you can see this seasonal increase also in the controls. The controls didn't have any oysters. The same happened also in 2019. What we did observe though is that the sediments, what we did observe though is that the bottom cages and the floating bags, the two techniques that we used for oyster culturing, had either similar levels or a bit higher levels compared to the control, specifically after the end of July again, which means that we had the potential two of the oyster treatments that we used somehow to contribute in nitrogen retention in the ecosystem. However, what we did find, and it was really interesting, is that one of the systems that we had, the oyster growth system, showed a total different trend compared to the control and compared to the rest of the oyster treatments that the sediments, the samples that we analyzed from the rest of the oyster treatments. So here also something important, here we are quantifying gene expression, meaning what? Meaning if this, how active or what levels this specific gene that describes this specific process was present during our sampling time. So it's not an actual number, but rather than an estimation of if the process is active, how active it is, and under which, let's say, treatments occurs. And I think that right now I will have to ask Dan to continue further.
Okay, so we're circling back to our big questions that we started off the talk with. There we go. And so how do we address these questions with the data that we've measured? Does aquaculture activity change nitrogen release from the sediments, the flux from the sediments? Yeah, we see in both years, we see an increased flux in all of the aquaculture treatments over what we saw at the control bare sediments. And we're seeing about a threefold change in August and September. The microbial communities are not changed. We see from our ITAG data that we had the same dots, the same representatives from the control site and the 3 oyster sites. There's no substantial difference between the change in community, but the activity does change. And we see this upregulation of especially genes associated with denitrification, and it coincides with our increase in our nitrogen release data in the last 2 months of the growing season. We drilled down further and looked at what activities are being enhanced as a result of aquaculture. And we see that Animox is enhanced, especially under the bottom cages, where we think we're dumping in so much organic matter that some of the ammonium is staying around and is available for Animox. We've also put in so much organic matter that we've drawn out all of the oxygen. Oxygen is going to be the first electron acceptor, the first thing used up to burn the organic matter that's being put in. And so by putting those oysters right on the bottom, the efficiency of that organic matter transport leads to this process being able to be expressed. DNRA was a little bit of a surprise for us. We see DNRA active under the floating bags and the oyster grow and even the control sediment. So this is a process that is natural in the environment. We do see slightly elevated rates in the floating bags and the bottom cages. And this is, again, a function of putting so much organic material into the sediments that we're starting to have this competition for nitrate. Well, the competition is between DNRA and denitrification. And one of the things that controls this competition is the amount of sulfide available. If sulfide gets high, greater than about 50 micromolar, DNRA has a competitive advantage over denitrification. And so we'll get a shift of moving our nutrient nitrogen to dinitrogen that can escape the environment back to ammonium, which can be stored in the environment. And so this is a very critical shift. Why didn't we see this in the oyster grow cages? Well, we think it has something to do with the physics of the oyster grow cages in the environment. These are big, heavy cages on these big pontoon floats. And as they bounce up and down in the water column, we think they're forcing the overlying oxygenated water down into the sediments, ventilating that top part of the sediments. Adding oxygen to the sediments will not only oxidize the ammonia that's there, but it also oxidizes any sulfide that's there. And so if we start repressing the sulfide, we go back to denitrification. And we think that's what we observed in this setting. So is there enough nitrogen being released in the sediments to impact our water quality management planning? Well, let's look at the ratios. What we typically do is we take the total biomass of oyster generated, we multiply it by some multiplier, usually 0.03%, I think is the multiplier they usually use. And we say we take out that much nitrogen by taking the oysters out of the ecosystem and putting them on the market. Well, for a system that was considered nearby to our experimental setup, they were looking at taking out about 14 kilograms of nitrogen per year. From our calculations, the amount of nitrogen that would have been removed from the footprint of our system would have been about 10% of the total nitrogen removed. So that's a significant amount of nitrogen being removed. So instead of 14 kilograms of nitrogen, maybe you're getting 15.5 kilograms of nitrogen. That's a significant impact on the management plan of that ecosystem. And that's really all happening over the last 2 months of the growing season. So our implications for management, it all comes down to your limitations. What gear do you want to choose given your limitations, and what is your ultimate goal? We're going to talk about the goal for nitrogen removal. We're not trying to grow pretty oysters for the market, although we can do that as well. We're just going to talk about nitrogen removal for today. Our gear choice depends on how much energy you want to spend to maintain your system. It's really easy to flip the light floating bags. It's a little bit more effort to flip the heavy bottom cages. We can talk about cost, hydrodynamic regime. Bottom cages are more expensive than the floating bags, but they also stand up to currents and tides much better than the floating bags. But from a nitrogen removal standpoint, bottom cages gave us the best nitrogen removal. We just have to be careful about the conditions of the sediments. If we put too much organic matter in, we'll generate too much sulfide, and we might shift that ecosystem back to ammonium storage through DNRA. So we need to know a little bit about the sediments that we're going to put these cages on. Well, in order to know what's going on in the sediments, we can simply make a couple of measurements. We can measure the carbon content of the sediments. That's a fairly easy measurement to make. We can make that at most of our county labs. It's simply taking our sediments, drying them, and running on an elemental analyzer. We can also measure the amount of sulfide in our sediments before, during, and maybe after our aquaculture efforts. What I have pictured here is a commercial test kit for sulfide. It's relatively cheap, for $200 for, I think it's 50 assays. And it's simply a colorimetric assay. It tells you how much sulfide is available in those pore waters. And if we have more than about 2 ppm or 50 micromolar, then we're going to have a system that's going to tend to store nitrogen as ammonia rather than remove it as dinitrogen. Again, these are very routine measurements to be able to do. If we do have a system that is getting too organic rich and too sulfide rich, perhaps what we need to do is rotate our crops, bring in different animals, move them to a slightly different area in the bay, but it becomes a management strategy that we can exploit. So in conclusion, all three systems increase the amount of nitrogen removed compared to the control sediments that were under the same hydrographic conditions. We didn't change the bacterial structure of the sediment community. That's important because we're not pushing the community to something that it wasn't already. We're just enhancing one feature of that community. We saw genes consistent to explain the nitrogen removal. So we have both the activity and the mechanism that drives that activity. And we have the warning that if we push these systems too hard, we might shoot ourselves in the foot. We might drive the systems to store nitrogen as ammonia rather than remove it as dinitrogen. So with that, I'd like to thank you for your attention. I'd like to take the opportunity to thank all of my colleagues and our funding agency. So this is a collaboration between myself at Woods Hole, Vivian and Jenny Edgecombe at Woods Hole. I'm at Stonehill. The Town of Falmouth, who managed our oysters for us. They've really been setting the bar in how to do this municipality oyster remediation projects. And Tonna and her group at Wacoit Bay for both support and all the things that the reserve system offers for us. And of course, last but not least, our funding agency. We're funded by the Science Collaborative, which enabled us to do all of this work. So with that, I will give it back over to Lynn, and I think we all have questions.
Yeah, absolutely. Thanks, everybody. So we are starting to get some questions. We just switch to the next slide here, just some quick instructions here. I encourage you to put your questions right into the Q&A feature and not the chat. It'll just make it a little bit easier for us. And then you can also upvote questions that you're also interested in to make sure we have time to address those ones that are most of interest to this group. And then anything that doesn't get answered today, we will put it in the summary document that will be shared after this webinar. So we are getting some questions. I'm going to jump right in. This first one, I think Tonna-Marie, you're probably the best position to answer. It was the question that came in early on about somebody who wanted to better understand the relative importance of the different sources of nitrogen to your coastal waters. And if you could say anything else about regulatory programs as well.
Thank you, Lynn. And that's a great question because it gets to the heart of where communities would direct their management efforts. So for us on Cape Cod, the bulk of the nitrogen that we're talking about here that's impacting the coastal bays and estuaries comes from those septic systems. So it's coming from that source. Other sources would be from the fertilizer use. And then there's atmospheric deposition of nitrogen as well. But when you look at the whole budget of that, most of it is coming from septic systems. So that is where most of the management efforts has been directed. And yes, there is the state level effort and county level effort to address regulations for nitrogen loading. And our Massachusetts Department of Environmental Protection is covering that. Towns have been developed TMDLs, total maximum daily loads of nitrogen that they're trying to achieve. And most communities are then developing these comprehensive water quality management plans where they're looking at what percentage of nitrogen could they remove using X method, B method, and sort of put that composite together. So the regulations are in place driving that whole process.
Thanks, Tonna-Marie. And there's one other kind of big picture question maybe you could just respond to. Somebody wants to know, does the quality of the oyster itself change as it's absorbing, so much nitrogen through its food? And will the oysters be safe to eat?
Dan, you want to take that, or would you like me to take it? Okay, Dan's going to take it.
Okay.
I have to find my meat button. Okay. The oysters are safe to eat. So all of the oysters that we grew were histology checked for pathogens and then relayed out to local sand flats. where they can be harvested from the recreational or commercial fishermen. But they are safe to eat. This is what oysters normally do. There's nothing special about it. Was there a difference as they took in the nitrogen? No, the oyster grows the same, but there's a difference between the oysters grown in, say, the floating bag versus the bottom cages. What our colleagues at Falmouth told us was that the oysters in the bottom cages were not as pretty, and that has an impact on their marketability in the fish market. So if you want really pretty oysters, you go with the floating bag because they're not messed up, they're not fouled as much by being near the sediments. If you want lots of oyster, you go with the bottom cages. So we saw better growth in the bottom cages, but the oysters weren't quite as pretty. We saw less growth in the floating bags, but they were beautiful oysters. And then the oyster grow also was sort of the middle. There were beautiful oysters and a little bit more bigger.
Okay, thanks, Dan. So here's a question from the Q&A console here. Wondering if you took any cores outside of the immediate oyster farm area. And I think they're interested in particular if this nitrogen removal impact of the aquaculture itself extends beyond, and to what extent it extends beyond the immediate cages.
So,
Yeah. Go ahead, Vivian.
No, that's fine.
So we sampled the control site as a bare sediment site. So that's outside the influence of the aquaculture systems. We did not do sort of a transect from the center of one system going out to see how big of a cloud there is around it. That is something we've talked about doing at some of the deeper sites. These sites are extremely shallow. These are all walkable. So these at their deepest are about 1.5 meters. So I can walk the whole thing. And have walked the whole thing. So I hope that answers the question.
So also there is available literature that actually points out that in sites or in areas where actually you have like many years of aquaculturing happening. So when people, they try to do what it is called chronosequencing, being able to identify which processes of nitrogen are enhanced in the presence or the absence of oysters, what they have found is that denitrification happens in the sediment even when the oysters are removed from the site for a year. So basically, it's like when you have the oysters, you train the sediment, you train bacteria to remove nitrate faster and to cooperate with that without the oysters even being present, even after a year. They continue denitrifying fast. So this is interesting because these results come after after the sediment technically should be back in the normal condition that it used to be before the oysters, but the communities are denitrifying pretty fast. So yes, the phenomenon is extended long term, not only to the sites but to the bay in general.
Okay, that's fascinating. So I got a policy question here. This might be for you, Tonna-Marie. Wondering if the state has a nutrient trading program whereby aquaculture farms can sell credits for nitrogen sources, such as to wastewater treatment plants, with potential for revenue streams for growers.
Yes, very interesting question. And the whole idea of nutrient trading is one that has been batted around for a long time on the Cape. There's been a lot of discussion with that, but there isn't one in place right now. The communities are really focused on trying to develop these comprehensive plans and sort of figure out what option, what place aquaculture could play and what credit they would get from regulators for using that particular option. So here's where a study like this is going to help regulators understand how much investment or how much trust can you put in a method like this to be able to achieve the limits that you need to get to. And aquaculture isn't the only one option that towns are looking at. They're looking at a suite of different options that they call non-traditional approaches along with soaring. So I'll stop there.
Okay. So I think this is maybe a question more for Dan about the sampling methods. Wondering how you extracted pore water from the sediment cores for analysis, and how did you decide on sampling depth for pore water nutrients?
So for the pore waters, we focus on the upper 3 centimeters of the sediment column. We extracted them of couple different ways. We centrifuged out poor waters, but we also did a calcium chloride wash so that we can get out all the extractable ammonium from the sediment column. So ammonium will stick to the sediment particles just ionically. So to get true ammonium readings, we needed to do a extraction process. So we're focused on the upper 3 centimeters, and that's how we got all of our ammonium data.
Okay. So here's kind of another sampling design question. Someone's wondering if there could be some bias in the results. For example, the currents may carry, bouncing around a little bit here, the currents may carry water back and forth. And how would you quantify and remove that impact of the currents moving around some of the chemistry?
So the currents can push nutrients back and forth, push particles back and forth. That's really what we're concerned about, back and forth between them. So one way we tried to attenuate that was we spread them out ever so slightly. Well, not ever so slightly, but a good distance. So again, we're in at high tide about 1.5 meters of water on the back end of on the deep end of the aquaculture systems, and then there's 5 meters between each type. And so you'd have to have quite a current ripping through there to push particles that far. The way the bay is structured, it's a shallow bay all the way through. At its maximum depth, I think it's 5 meters deep. And that's way out in the middle of the bay. And so we're way up in the head. We have mostly wind-driven currents. Most of the wind-driven currents are going to be onshore and then across once they hit the shore. So that's how we're trying to moderate our transport, our vective transport of particles and nutrients between the sites. We're also comparing everything back to that control site so that we're normalizing to that control site. So whatever is washed back and forth would also be washed back and forth to the control site.
Thanks for that. We did try and do different types of sediment traps with varying degrees of success. Sediment traps were a bit challenging because of the shallow water and because when the blue crabs come in, they destroy everything. So yeah, we had some sampler faults due to crab violence, I guess. Yeah, and maybe that actually kind of flows into a question here. I'm going to jump around a little bit here about the, is there any evidence to suggest that other prolific but less desirable invasive species, such as zebra mussels, could also contribute to denitrification? So maybe tenuous connection there between other invertebrates, but.
The answer is I don't know. I don't, again, I'm the chemist. So the way I think about this is how do you get the nitrogen and the organic matter to the sediments? Could we do this with other shellfish? Absolutely. Could we do this with things besides shellfish? Sure. We have to get the nitrogen into that anaerobic zone with just the right amount, with not too much sulfide that we push it to DNRA, which will just store the ammonia temporarily in the sediment column. You could do it with invasive species.
The only 2 comments here. So the first thing that we have to take into consideration is that denitrification as a process contributes to nitrogen removal on, let's say, 30% of the ecosystem. The rest of the 70% that it is removed from an ecosystem via the oysters is through their biomass. So basically, because the oysters are growing and they are uptaking all the nutrients from the water, they include it in their biomass. So definitely, by having any type of bivalve that actually can filter feeds on nutrients, it's going to remove nitrogen from the water column. Considering how good the denitrification is going to be, or how well it's going to be triggered underneath this species, it's something that it is, it's questionable. But as I said, don't forget that the vast majority of the nitrogen that it is removed from the ecosystem is via the biomass that accumulates in the organism.
So all the better if it's a species that's getting harvested.
So if we have a species that accumulates a lot of nitrogen and at the same time triggers a lot of denitrification, it's the best combination that we can have.
Right. Yeah. And zebra mussels aren't typically harvested and eaten the way oysters are.
Yep.
No, and oysters are a natural part of our ecosystem. They
Yes.
used to be exceedingly like navigation hazard common. So they should be in that system.
Absolutely.
That's one of the drivers. It's not just their market value, but they should also be
Friendly to the ecosystem that you grow. Yes.
Okay. So I think this is a question, this is from Jeffrey Cornwell. I think maybe thinking about a different ecosystem, slightly different ecosystem. He says that he sees attenuation of denitrification with illumination, whereby nitrifiers lose out to autotrophs. And he's wondering, do you think this might diminish denitrification?
So in our system, the nitrate is already coming out of the groundwater system. So with illumination, I think what he's talking about, illumination, you're going to stimulate the phytoplankton. They're going to start assimilating that nitrogen faster than the nitrifiers would be. Nitrifiers move nitrogen from ammonium to nitrate, but we already have it in that nitrate pool. And we're trying to collect it and put it into the sediments where it will be removed. If we're talking about benthic, so phytoplanktons that are in the surface of the sediments, we illuminate those, which is entirely likely in 1.5 meters of water. Ammonium that's released and might get taken away by the phytoplankton before it can be moved to nitrate and removed. That's entirely possible. We don't have a number on that. We measured net nitrogen flux. So if it's happening, it was happening in our cores.
Also, don't forget that when we are having this type of processes, denitrification, sometimes, depending on the condition, is coupled with nitrification, because basically you need somebody to be active, to provide the nitrate, then for somebody else to use it in order not to denitrify. So there is a very, let's say, thin line there. Sometimes, you know, you can find coupled these two processes. So I will stick with the answer that Dan gave.
Okay, sounds good. I think we probably can do maybe one more quick one here. Someone's asking, can you imagine a scenario whereby too many oysters could lead to a worsening of water quality?
So go ahead, Vivian.
So here, from what we find, at least from what we found and what we compared towards the published literature. So all these, so the thing is that when we are thinking about an ecosystem, we think, oh, okay, nitrogen cycling happens here. That means that the sulfur cycle that happens right here, that has nothing to do with what we are studying, which is not correct, because, you know, all these processes are there's such a great crosstalk between all these processes. So you have to take into account all the parameters when you want to study a specific cycling like nitrogen. So in our case, we saw that literally presence of high hydrogen sulfide in the sediment definitely is going to have a result that we don't like, that we wouldn't like to trigger. So in this case, we have to take into consideration, first of all, the local geochemistry, meaning the local sediments from where we want to set up the oysters, how do they behave? We have to monitor before we do any, let's say, further step regarding aquaculture. And then the thing is, okay, since we have the option to pick up the safest way of culturing the oysters, these are things that we should take into consideration. Meaning, for example, if we had, let's say, a very big surface area that had oyster grow, I don't believe that we would push the system toward the NRA, just because the system by itself, the treatment by itself, aerates the sediment. So basically breaks down all the anaerobic, all the conditions that might be anaerobic and thus lead to sulfide accumulation and thus lead to nitrogen retention overall.
Okay. Well, thank you all. I think we're at the top of the hour here, so I'm gonna cut us off. But like I said, we are gonna gather up any questions that weren't answered, and this team has agreed to help us get answers to them, and we'll put them in the summary. So I guess I want to close with just a big thank you to all three of you for the time you put into this presentation and the really thoughtful presentation of some really technical material. And you can see some really technical questions too, right? I think you sparked some interest here in this group.
Yep.
And I'll put in one quick plug for our next webinar coming up in this series is on June 29th. It's going to be a panel style webinar featuring folks who are participating in the Margaret A. Davidson Fellowship, which is a relatively new fellowship program that puts graduate students doing collaborative science projects in partnership with the reserves. So similar to what we've been talking about today, but specifically for graduate students. And they're going to be accepting applications starting in June. So it's a timely webinar, so check that out. So again, thank you to everybody who joined us today, and big thank you to our presenters. And we'll sign off there. Have a great afternoon.
Thank you, Lynn.
Thank
Thank you, Megan.
Thank you.
you.